Welcome to W&J Science Talk Radio. Today we have 4 students from Dr. Kilgore's Bio 111 class to talk to us about their experiment with soil carbon content in areas of mushroom density. We have Jen Shear, Abby Pease, Annalise Rice, and Ryder Doss joining us on the show today.
Have you ever wondered why mushrooms keep growing in your yard? Well, we have an answer for you.
You have a large amount of carbon in your soil.
How do you fix that?
You can't, but it isn't necessarily a bad thing.
As it turns out, mushrooms fix the carbon in the soil and use it to grow. This carbon can be used by other organisms after the mushrooms are gone.
We discovered this by walking around campus, trying to find forms of biodiversity, and came across mushrooms growing in 3 patches throughout campus.
These patches were present at Olin, the gazebo by Mellon, and outside of Mellon on Washington and Jefferson College campus.
We concluded that there had to be a reason these mushrooms were growing in these set locations instead of everywhere.
We decided to take 3 soil samples inside each patch so we could test the carbon content in the soil and develop an average for each location.
We gathered the needed materials and researched a way to measure the amount of carbon present in the soil.
With the use of loss on ignition method, we were able to measure the carbon in the soil and compare the sites.
We then questioned if the carbon content differs outside of the mushroom patch and decided to take 3 samples of increasing distance away from the patch.
All 18 samples were taken and then ready to be tested in the lab.
The chemistry lab was open for us to do our testing and measurements.
We started by picking out all the rocks, roots, sticks, and leaves from the soil samples to get the purest form of the soil in crushed clumps to make it the same consistency.
We placed these samples in an oven set to 105 degrees Celsius for 24 hours in order to remove the water from the samples.
The samples were weighed before and after heating them in an oven to see the amount of carbon lost.
The mass of the 3 on-site samples at each location were averaged to represent the carbon content at each of the sites.
A 95% confidence interval was used to determine differences between sites.
The graph showed that it is inconclusive if soil carbon content is dependent on distance from the mushroom patch.
The gazebo on-site average was much higher than the other on-site averages.
The gazebo showed a decreasing trend, while the others had a fairly constant trend.
The gazebo had the highest carbon content in the soil, and that could correlate to the very large density of mushrooms present at this location.
The gazebo results supported our hypothesis, but the trend seen in the other sites shows that the gazebo trend may not be true for all cases.
Overall, the data was inconclusive.
So, although there may be mushrooms in your yard now, they have a short life cycle.
The mushroom tops are the reproductive organs of the mushroom and are used to make spores to create more mushrooms.
After the mushrooms are gone, the hyphae are still present in the soil.
Hyphae are string-like ribbons that create chemicals to break down and decompose food for the mushroom.
Mushrooms aren't bad and will help your yard in the future by making the carbon readily available to other organisms and plants to use.
We would like to thank Dr. Kilgore, Rebecca Wilkes, Dr. Polvani, and all others who helped us make this project a success. Wow, I never knew so much more went on with mushrooms than what's above the surface. I hope you guys learned a lot and will tune in next time for W&J Science Talk Radio.